All right. Of course. Okay. So we're going to get through the material pretty quick here because there is very, very little math. So that means I basically just tell you these things and there you go. There's actually fairly low cognitive load stuff. There's actually almost no new information. It's really just information we already have taught you being applied to biological systems now. Okay? That's why we did all that work before, right? Yeah. All right, so tertiary structural interactions. These are the sort of interactions that are going to keep your tertiary structure in that three-dimensional arrangement. Okay? And so we have nonpolar interactions. These are going to be the nonpolar amino acid side chains. They're going to be repelled by the aqueous environment, and so they're going to be squeezed together so that they then have those nonpolar interactions, the London dispersion forces, on those environments. Now, you will note that whenever we're talking about a protein that's exposed or an enzyme that's exposed to aqueous solutions, like your blood, your cell interior environment, not your cell membranes, these are going to be inside of the protein. If you're exposed to the nonpolar environments like a cell membrane, they're going to be on the outside. Okay? Polar interactions, these are the polar ones, obviously. So they're going to be the polar amino acid side chains. These are ones that interact with water, of course. They're going to interact with each other as well through dipole-dipole interactions. You can also interact with ion-dipole interactions and hydrogen bonding interactions. And so most of them have hydrogen bonding. There are a few that are dipole-dipole interactions or dipole hydrogen bond acceptors. And then there's always the ion dipole that's possible. Okay, and in fact, happens quite a bit. In fact, you guys know you need like magnesium in your diet, right? And potassium in your diet. Yeah, so magnesium especially is very common to make ion dipole interactions, and it actually serves to help structure your proteins quite a bit. Actually, it's one of the reasons why it's so important in your diet. You probably have heard like the commercial for Magnesium Breakthrough, right? Yeah, they're not wrong. Magnesium is like important for almost everything because it's such a ubiquitous metal in your protein structures. Salt bridges. These are going to be called ionic interactions. This is where you have an ion matching with an ion. They're pretty strong interactions though, but they can be interrupted. Okay. This can be things like your acidic and basic amino acid side chains, and that's why we subdivide those out on those categories, right? So we have our nonpolar category, our polar neutral category, and then our polar acidic basic category, right? Which also could mean positive, negative. So these are going to be opposite charges. They're going to attract. They're going to stabilize ionic interactions. Whenever we have these stabilizing ionic interactions, we're going to call those salt bridges because they're side chains that overlap, making those ionic, and they look a lot like a bridge, right? You know the gated bridges that you see in cities? Like that. Disulfide bonds, these are actually covalent bonds, okay? So they're actually linking covalent bonds here. And this happens when you have two thiols. They're going to react through an oxidation reaction. This will be losing those hydrogens. And they're going to form these disulfide bonds. So a sulfur bonded to an amino acid over here, covalent bond, and then another amino acid on the other side. And they'll be, far enough apart in the chain that they can curl back in. Okay? In case you're wondering, these are the sort of interactions that cause your hair to curl. So if you have curly hair, these are the interactions you're seeing, actually. Okay? And in fact, if you get hair straightener, the reason it smells like it does is it's actually an agent. So it's going to cause... God, this thing is just so... It's going to be an agent that's actually going to oxidize, reduce these bonds here. So it's a reducing agent that's going to reduce these bonds down to the HS's, cause those to break, and that's going to relieve that kink in your hair, making it straight. Okay? It's also why you usually need to provide heat to do it as well, because you need the energy to break those bonds. Now, this is actually this protein backbone right here. Right here we have the thiols. Here we have the disulfide linkage. And so this is an oxidation to get the disulfide linkage, a reduction to go back to the thiols. Hey, we got our redox reactions that we were talking about, don't we? It's like everything's coming and building up to something, which is you all, living organisms. Woo. Good stuff. All right, now, this right here is going to be, again, your tertiary structure. This would be a globular proteins are going to fold into a very compact spherical type of shape, and this would be an example of a globular protein. You'll see that here's the N-terminus, right here's the C-terminus. That's the side with the carboxylic acid, obviously. This is the side with the amine, right? Okay. So, in this case, because it's going to be a water-soluble one that's going to interact with water, typically proteins don't actually become water-soluble. They're just too big. So they're going to just be suspended in aqueous solutions. In fact, if you were to just sit around and have nothing else happen to you, you will have your proteins and blood cells, etc., all congeal down. And that's what happens to a corpse, actually. That's why it gets really gross. Everything congeals down and sticks to stuff, right? Yeah. You guys have seen roadkill on the side of the road, right? Like that. Human can also be roadkill. Okay. Hopefully nobody picks it up to eat. I've been seeing since I moved to Tennessee. Okay. So again, globular proteins. We're going to have our polar interactions exposed on the outside, our non-polar in the center, helping to hold that together. Okay. Right here, we actually see fibrous proteins. These are going to be long thread-like structures. They'll be aligned in helices, usually braided helices, to create these long, durable structures. They tend to be insoluble in water because, for the most part, you don't want your structures to dissolve. That seems counterproductive, right? Especially since we're mostly composed of water. No Wicked Witch of the West here. Okay. And so right here, obviously, we see hair. You can see that is the keratin. This is the coiled alpha coils where the keratin coil together to form these strands of hair. Note that you're actually looking at, whenever you look at hair, you're actually looking at very long molecules, polymers, that are just continuously lengthening out. Okay? And at some point, they actually do get too weak and they break, because as they go out from you, they're going to get weaker and weaker. Okay? This right here then is collagen. You may know what that is. It's your skin. So it makes up your skin. And so that's actually the fibrous protein that's going to make your skin. Note that you're actually quite durable to puncturing, right? Compared to most structures. You don't actually break, do you? You take the puncture and then you mostly seal up. Okay? And one of the major damage points and the most common method of things killing you happens to be puncturing. Yeah. So it's a little bit like a bulletproof vest over you, just not quite as strong as the polymers that humans can make, right? Partially because those would be incredibly energy intensive to make if you were to actually try to make that as a human. So quaternary structures. Now, quaternary structures is going to describe the interactions of two or more polypeptide chains. This is going to form a single biologically active protein. And so what you're looking at as quaternary structures is tertiary structures added together. Okay? So when you're looking at a tertiary structure, it's only one chain. And you'll see that this right here is just one chain folded up, right? Everybody see it okay? When you get to a quaternary structure, this can be multiple of those chains added together. Okay? And so, for example, hemoglobin actually has 4 subunits. So your hemoglobin actually has 4 subunits, 4 chains that are brought together. That forms your hemoglobin. Okay? And that would give you your quaternary structure. Now, the individual polypeptide chains or subunits are going to be held together by those same interactions that we had seen stabilizing the tertiary structure of your single protein. And so what you'll end up seeing is the difference between a quaternary and tertiary structure is actually pretty minimal, surprisingly so. Okay? The only real difference is we had one chain, now we have two or more chains. Not all biologically active proteins have quaternary structures. Some of them, in fact, a pretty good number, are actually made of single chains. Okay, myoglobin is a single chain, only has tertiary structure. Hemoglobin is very similar to myoglobin and has a quaternary structure. It has actually 4 of them. Okay, you don't have to have 4 to be quaternary though. Just 2 or more chains. So again, this is a summary of what we just went over. This is a graphical summary of what we went over. So this would be primary structure, the order they're in. Secondary structure is the individual motifs, patterns, kind of like your organelles or the parts of a car, right? And then you add those together, the secondary structures, and you're going to get your tertiary structure, which is a single peptide, polypeptide, that chain that has made that 3-dimensional structure. And then if you have more than one of these, like in, for example, this would be a hemoglobin right here, actually. That would be your quaternary structure. Okay. By the way, those are the hemes right there. Very cartoony looking one, but there you go, hemoglobin. All right. So let's talk about the denaturation of these protein structures. Now, notice that those stabilizing attractive forces in the secondary, tertiary, quaternary structures, those were mostly pretty weak forces. Those are your normal intermolecular structures, right? And we know hydrogen bonds are actually pretty easy to boil because we boil them all the time, right? We boil water. And so we know that splitting those up is not that difficult, is it? And so when you cook something, what you're doing, you're denaturing that protein, okay? Or you've denatured that protein is what you've done. And so, again, your primary structure is not going to change. Your overall protein primary structure doesn't change at all unless you burn your food, okay? What you're actually doing is you're causing these intermolecular forces to split up, and that unravels the protein structure, and then that gives you only the primary structure. You don't have the secondary, you don't have the tertiary, you don't have the quaternary structures anymore, okay? And that makes it so that it loses all of its biological activity. It no longer does what that enzyme used to do. The protein no longer does what the protein used to do because it's lost the form that would give it its function. Okay? It's kind of like if you were to take one of those grabber claws and you were to melt it down, you still have the plastic, right? And it'd still be that plastic. Wouldn't grab anything though anymore, would it? Okay? Same thing here. It no longer will be able to perform its biological function. And that's actually what you're seeing here. So these are the denatured proteins right here. And these ones right here, the non-denatured ones. You can actually see the difference in that case, can't you? Okay. Everybody's comfortable with that? Any questions? No? Is heat the only way you can cook stuff? No, you can do many other things, right? Like pickling, or you can actually. Have you guys ever seen the videos where they make slap cooking? Okay, this is kind of funny. When you slap something, you're transferring kinetic energy to it, right? And so people have made machines that essentially slap something like 10,000 times over the course of 30 minutes, and it actually will cook stuff. So you can look at one of those videos if you want to see something amusing, okay? But there are actually many, many different ways you can cook stuff. As long as you can break the overall 3-dimensional structures, you're usually going to accomplish cooking, at least for proteins, okay? Now, without stabilizing these interactions that are necessary to maintain its 3-dimensional structure, a protein will unfold into a shapeless string of amino acids. The conditions to denature a protein are mild when you compare to like a protein hydrolysis to actually break it apart and no longer have that primary structure. Those covalent bonds in the peptide linkages are quite a bit stronger, actually. And so this usually requires very high temperatures or harsh chemical conditions to break apart that primary structure. And so if you have heat above 50 degrees Celsius, this will slowly cook things. You would not normally try to cook it like that because it would take hours and hours and hours. Very long time. It'd be like cooking on one of those steam plates that they serve in the cafeteria. Would it cook stuff? Yes. Would you want to wait for it? No. Typically, we cook at something like 80 degrees Celsius. Okay? That'd be more the temperatures we would cook at. Something close to boiling, right? Acids, bases, ionic compounds can all accomplish some degree of breaking these apart. And so this is one of the reasons like you can use this to decontaminate and kill microorganisms. So for example, lactic acid from bacteria will denature the milk proteins. That's actually how we get yogurts and cheeses, isn't it? Okay. And so that's an example right there. Reducing agents can actually separate those disulfide bonds. And so the thiols, these are used as hair straightening and permanent waves because you can actually modify the disulfide bonding and then give different hair structures, right? Detergents are actually very good at disrupting hydrophobic interactions. This can disrupt proteins, but typically the ones that you're going after when you do this is membrane proteins specifically. Okay. Heavy metal ions are very good at breaking disulfide bonds, but also good at salt bridges as well. And so, for example, mercury and lead are very easy to cause poisoning, and you can do even very small concentrations of them will give some degree of poisoning to people. And that's because they're very good at inserting themselves into disulfide bonds especially, but also salt bridges. The disulfide bonds bond very strongly to mercury, by the way. So it's actually incredibly difficult to get rid of mercury once you've been poisoned. Okay. Mechanical agitation can be used. This is probably, have you guys taken, has anybody taken micro or going to take micro biology? Yeah. So I have some of my classes nurses. This is probably the one you guys would use if you're going to break apart something. Typically, it looks a bit like a plunger in like a test tube. And you go, or you can use a little blender looking thing. Okay. And so essentially, you just break it apart with physical force, right? And this often works really well with hydrogen bonds and London dispersion forces. Some examples of this would actually be whipped cream and meringue that's made from egg whites, obviously. So you take egg whites and you whip it really fast. And that's why it quote unquote cooks, is you're actually mechanically breaking that tertiary and quaternary structures. Okay. And so you can do that as well. That's why the slap cooking also works, by the way. Same reason. Though I don't think anybody's going to sit there slapping a chicken to cook it. Hopefully not. No? Okay, so this is actually hair relaxing and perming. This is going to involve, of course, your protein denaturation. It's going to be where you take these disulfide linkages. These are what cause the curling in people's hair. And you're going to break those disulfide linkages. Right here is actually one of the denaturation agents. And then what you can do is then reestablish them in the shape you want. Okay? And so you then oxidize them back with an oxidizing agent. Typically something like hydrogen peroxide, but you can actually use several different ones. Okay? Hydrogen peroxide tends to be the more common. And that's that right there. Can everybody kind of see how that would work? So normally the linkages are downwards throughout the structure of your hair, and that gives the really curly hair. If you only do this occasionally, that gives you a slight curl, right? Because it just bends it a little bit. This then would be like a wave that you're establishing in the hair, or a more straight hairstyle. Okay? And so what you do then is you break the linkages, you then smooth it out to what you want it to be shaped as. You kind of hold it in place, usually with some tin foil or something, right? Everybody's kind of seen this being done. Okay. You can go the other way too, by the way, and give it curl by holding it in place with tin foil or a curler, right? And then you're going to put an oxidizing agent in there. That'll reestablish the disulfide linkages into the shape that you want it to be in. Okay? It will eventually actually break because you're naturally going to be reduced and oxidized when you're in contact with the atmosphere. Okay? So these do fade over time, right? That making sense to everybody? Okay. Cool. All right, so these are some different protein functions of various different types of proteins. So right here we actually have insulin. This is the messenger protein. We have the insulin receptor. That's a receptor protein. We have transport proteins, for example, a glucose transport or hemoglobin. These are both transporters. We have storage, and so they're going to store nutrients that would otherwise fade from your body. For example, casein stores proteins in milk. Ferritin stores iron in cells. Part of the reason you need to do this is because you continuously have to intake and expel water from your body. Things that are water soluble don't stick around in your body. And so, for example, vitamin C, we don't store that well. And so you have to have new vitamin C every 2 to 3 days because you're going to urinate out all your vitamin C over time. Okay. Myosin and actin are going to make up thick and thin filaments in muscle tissues. And so these are for contraction. We have antibodies, of course, which are protection. We have structure, things like collagen. These are going to form things like the ligaments, tendons, skin, nails, horns. All of those are formed from collagen. Okay. We have actin, of course, and that's the supportive structures of our cells. Those are the cytoskeletons, if you ever take some biology. So we have the catalysts. Those are going to be pepsin, for example. Pepsin's actually what's responsible for breaking peptide bonds in our stomach during digestion. It's the sort of first steps of that process. Anybody know what the first step of breaking starch down is? Where that's located? Yeah. That's why your hands always get sticky. It's because you're always getting saliva on them. Because people touch their mouths all the time. Yeah. Okay. And so proteins can act as, of course, messengers, receptors, and transporters. And so a hormone, this can be a chemical that's sometimes a peptide or protein, but doesn't have to be. Remember, quite a few of them are going to be lipid-derived structures. So this is going to be one part of the body that can affect another part of the body, and it's going to be a widespread cascading messenger. You need very little hormone to get a huge effect. Okay? By the way, be very careful if you ever inject hormones into your body, whether that's on your own or a doctor doing it for you. Yeah. Yeah. No, don't do wrong one. So the receptors, they're going to be proteins that face the outer surface of a cell. They're going to bind to a hormone or other messenger. They're going to trigger a signal that's inside the cell. And so what you get is actually an amplifying effect. Anybody know what an amplifier is, by the way? So you put a signal in, it then amplifies it to get better signal on the other side, right? For example, like your stereo or your earbuds have amplifiers where they take the signal coming out of your phone and then they amplify that signal so you get a bigger sound. Okay? At least good ones do. Bad ones do not. Right. So a transporter is going to be an integral membrane protein. It's going to span the phospholipid bilayer. And right here we actually see some receptor right here for insulin. And this one right here is actually a glucose transporter. Glucose is too polar and too large to go through the cell membrane directly. And so this helps transport it across the cell membrane. All right. So this right here is going to be hemoglobin. This is your oxygen transporter in your body. There's 4 subunits for the hemoglobin. We can see these as the 2 reds and 2 blues. And that's because those 4 subunits are actually 2 of the same subunit and 2 of the other same subunit. Okay? Each subunit is going to have a heme prosthetic group. That's this group right here. See the green with the little yellow ball on it? These are actually what bind the irons in your hemoglobin. And so that iron 2 plus is going to bond oxygen. And it actually looks a bit like a little trampoline with the metal sitting on it. Okay? And so it's going to bond oxygens. As you bond oxygens, these actually get better at bonding oxygens. And so they actually suck up a bunch of oxygen, and then they get pushed along in your blood. They get to a low oxygen environment, and oxygens start coming off. And they actually get better at losing oxygens once the oxygens start coming off. And so they then come off in your extremities. Right. So one of these hemoglobins actually transports up to 4 oxygens. For the most part, they're going to end up with 4 oxygens on them in your lungs. Not all of them will, but the vast majority. The binding of O2 in the hemoglobin is actually going to induce a conformational change. What you'll see is it actually is like this most of the time, that heme. And then when an oxygen comes in, it actually goes like that. And it straightens out a bit. And that's why they actually get a little bit better, is because it actually causes all of them to straighten out and bond easier. And then when you start removing oxygens, they start cascading like this, and they get worse and worse at bonding. Okay? And so it actually changes the entire structure when you start bonding oxygens. And this is actually the oxygen bound state right there. And you can actually see that difference, can't you? Because the hole right there makes it really easy to see. You can actually modify the process right here by putting it in acidic conditions. For example, acidic conditions make this oxygen bound state less stable and make this state more stable. Okay, so that's the oxygenated state, that's the deoxy state. So this is your antibodies. This is going to be your body's defense protein, of course. So when you have a foreign substance, these are going to be called antigens, like the bacteria that enters your body. Your immune system is going to produce proteins that are called antibodies. The antibodies are going to be consisting of 4 polypeptide subunits. This is what this one's the easy one to see, the 4 subunits. This one's a little more difficult to see because that's how they actually are. Nothing in your body is a straight line, right? All right. And so the secondary structure is going to contain beta-pleated sheets. These are the flat ribbons that you can see in some of these structures. They're going to be stacked tightly together. The quaternary structure is going to be held through disulfide bridges between those polypeptide chains. It's easier to see those up here, the disulfide bridges. The stem of the Y is similar in all antibodies, and these can be binded to receptors on a variety of cells in the body. The antibodies are going to bind the antigens at the top of each arm of the Y, and that's the place where you have the little identifiers that are going to identify the different antigens. Okay? And so basically what happens is your body creates sort of random structures on this end, and then it sees if it binds to it. And it keeps doing that until it finds something that binds to it. And then sometimes that binds really well, and you kill the invader. Congratulations. You're over the disease. And that gives you the antibody. And then you'll keep a certain level of those in your body at all times, so that in case that invader comes back. Okay? Sometimes the thing you make on the end here also happens to bind stuff in your own body. And then that is what is called an allergy. Okay? You have then an autoimmune disease or an allergy that's going to occur, depending on the seriousness of it. And then congratulations, that's going to be an issue from now on. This is why, for example, stuff that's very similar to our own structures, like bloodworm, it's very easy to get autoimmune diseases or allergies to. Okay? An autoimmune disease is just when this happens and continuously happens to you. And it can be fixed over time, but they're pretty tough to deal with. An allergen is when it only spikes when you immediately are having the substance in you, and your autoimmune response to it goes out of control. Okay? Okay. So let's talk about some of life's catalysts. Enzymes are going to be typically larger globular proteins. The globular proteins are the most common. Is going to be present in every cell of the body. Enzymes are going to oftentimes act as catalysts. It's not the only thing they do, but most of the stuff that you think of as being enzymes and doing stuff is probably catalysts. Compounds that accelerate reactions of metabolism but are not consumed or changed by these reactions, they are going to be catalysts. And an enzyme. So an enzyme cannot force a reaction to occur that would not normally occur. All it can do is lower the activation energy. So it doesn't make something that would not occur spontaneously. That's thermodynamics. It's not the speed of the reaction, right? And so, like, for example, if that reaction would not normally occur in your body, it doesn't matter how good of a catalyst you make, it's not going to happen, okay? But if it would occur but very slowly, then a catalyst is going to make it go faster. And it's going to make it go faster in both directions, okay? So any enzyme you make will make a reaction go faster in both directions. For example, you actually have a catalyst that converts CO2 to carbonic acid. What does that mean that catalyst also does? It converts carbonic acid to CO2. Yeah, speeds up both directions, right? And so that's actually how you transport CO2 in your body, right? You use that same enzyme in both places. Or very, very similar structures. Now, the tertiary structure of an enzyme is going to play an extremely important role in its function. And when you're talking about enzymes and we're talking about proteins, what they look like is directly linked to what its function is. Okay? There isn't a subdivide. Once you break what they look like, the tertiary structure, you also almost always break the function. Okay. So the enzyme's name usually is going to appear above or below the reaction arrows in these. You can see this reaction right here and the enzyme right there. That's hexokinase, right? The active site is going to be part of the enzyme where the catalyst occurs. And so active site is literally where the enzyme is active. The reactant is going to be referred to as a substrate. They're referred to as a substrate in these cases because they actually have to bind to the enzyme first. And so that's what a substrate is. That has to bind to the enzyme first. That's why it has a different terminology. The cofactors, these are going to be inorganic, usually, substances, and coenzymes are going to be small organic molecules that assist in the catalysis. This is a lot of the nutrients that you think of that you need in your diet are actually cofactors or coenzymes. They're things that need to be bound to the enzyme for it to function, or the enzyme needs as added material. They don't actually take part in the final product usually, though. They get reused. The active sites of hexokinase fits a D-glucose only, and this is due to the substrate specificity. Most enzymes will only take specific isomers of substrates because they themselves are isomers. Okay, they have a three-dimensional orientation, right? And that's because, remember, they all have those carbons where they have chirality, right? And so enzymes always have chirality in their structures. They're always isomers. And so they only work on a single isomer almost across the board. This right here is the D-glucose, and we can see it binds in a very specific orientation. If you were to mirror that, it would no longer fit in there and bind. Okay? Okay, so this is the enzyme substrate models that you would see. This is the lock and key model, and this is the induced fit model right here. And so the enzyme substrate complex is when you have the enzyme and substrate come together, and they are now bounded to each other. Okay? And this is why that terminology of substrate. It's because the reactant in these cases has to be bound first. Okay? All right, and so this is the lock and key model, where the substrate and the enzyme are going to fit together like a lock and a key, right? If you have the wrong key, it's not going to fit into that, right? Or you may have the where it fits, but it won't actually unlock, right? Because it's missing one of the key parts. Okay, one of the teeth, as it were. Is that making sense? And so that's the lock and key model. Now, this is actually not how almost any enzymes ever work, but it's a nice mental model. It's really simple. The model that usually is more correct on how an enzyme actually functions and work is what's called the induced fit model. And so when you put your enzyme and substrate together, your enzyme is not actually going to have the exact structure that's going to fit that substrate. It's going to be a little different. Okay? And when you put it together, what's going to happen is your substrate is going to slightly change your enzyme, and then they're going to fit together properly. Okay? So if you were to just look at your enzyme and go, does this fit? You can kind of see it, but not fully, right? Because you got to remember, these are not locks. They are not inorganic, inanimate objects, right? They are you. So how do you work when you pick something up or turn it? So it'd be like this, right? My hand is not perfectly fit this right now, does it? Pretty close. And so when it comes in, I'm going to change my structure. Now I'm going to perfectly fit this, and I can do transformations, right? And that's going to be the induced fit model. Okay? That is much, much, much closer to how an actual enzyme substrate's pretty accurate, actually. Okay? There's a few caveats and complications if you want to go into biochemistry, but for this level, plenty good. Right? Okay, and so this right here is, again, your enzyme and substrate are separate. The glucose comes in, it slots into that. It's going to then hydrogen bond the different parts of the structure. Your enzyme's then going to close around it because that hydrogen bonding is going to provide energy. And so it's going to want to hydrogen bond better, right? And so it's going to modify its structure to better fit. Okay. And then that's our enzyme substrate complex. Let's talk a little bit about reaction rates. So what you're going to see when you look at these reaction rates is that you're actually going to split this reaction into multiple steps. But each step ends up being lower energy. So let me ask you a question. Why the hell do we have stairs? Why do we have stairs? What if we got rid of them? The overall energy of you coming up to this floor doesn't change, does it? So like getting up to this floor would take the same energy whether you went up the stairs or whether you jumped directly up to the floor, right? So why do we have stairs? Can anybody make that jump? No, none of us can make that jump, right? And that's because we don't have enough kinetic energy, do we? And so what you can do in any system is you can subdivide the kinetic energy you need into steps. Okay? And that's what we're doing here. We're subdividing into steps, and that's going to make the reaction go far faster, right? Because a lot more of the molecules can make it over that barrier. Okay? Versus making it over this barrier, which is much larger. Right? That'd be like giant steps, right? Or a giant leap onto the next floor. Is that making sense? Okay, and so that right there is how an enzyme functions. So what we have first is the energy of our substrate, and whether we have an enzyme or not, that's going to be the same. Okay? But the first step for an enzyme is going to be binding the substrate to the enzyme. Okay? And so that's going to be this process right here. And we're making the enzyme substrate complex right there. Okay? And then what's going to happen is now the enzyme is going to be reducing the energy of that reaction, either by bringing what needs to react closer together so they collide with each other more efficiently, or by weakening some bond that needs to break. Okay? And so we get a lower energy barrier for the next, and that reaction can go much faster. Now again, the energy of the products, once we split off of that enzyme, they're going to be the exact same. Okay? Because they're the same product, same substrate for either way. All right? And so proximity, when the enzyme substrate forms, the substrates are going to be closer in proximity. They're going to be oriented more in the correct orientation. And so when they collide, more of those collisions will be meaningful. And that's going to greatly reduce this energy barrier. Okay? And that's how most enzymes actually work. The orientation is the other part that's going to help this out. And I had already mentioned that. But for example, if a reaction needs two of these to come together and you orient them so all their collisions are happening like this, that's going to make life a lot easier, right? Or another way of thinking about that is if you're playing sports and you're faced towards the ball, a lot easier to catch it than if you face this direction, right? Not going to be so easy to catch that, is it? That's all you're doing. You're orienting, aren't you? Bond energy. So when an enzyme interacts with its substrate to form the enzyme substrate complex, the bonds of the substrate molecules are going to be weakened. They're going to be strained. And so what that's going to do is it's going to make those bonds easier to break. This happens a lot in things like ATP. And this is one of the main uses of magnesium, actually, is you can use that to weaken the phosphate bonds. This is why the magnesium breakthrough. Increase energy with magnesium. There's some truth to that. If you're low on magnesium, obviously you'd be able to use your ATP more effectively if you have magnesium. But it's somewhat rare for people to be low on magnesium. That's actually would be kind of uncommon. But maybe you're. Substrate concentration. So the first step in enzyme catalyzed reaction is going to be formation of your enzyme substrate complex. If the amount of the enzyme remains unchanged, then an increase in substrate concentration will increase the enzyme activity, and it's going to do so up to a point where you've saturated all enzyme, where you're using all the enzyme you can. You can think of these a bit like queues at Disney World. Like if you don't have all the queues filled up, or queues at Walmart. If not all the queues are filled up, then you could open a new queue, you can go faster, right? But once all the queues are filled up, you're locked into that rate. You can't go any faster, can you? Yeah. So, if you increase the substrate beyond this point, it's not going to help. You're at your maximum activity. That's as fast as it's going to go. Okay. Substrate concentration. So at maximum activity, the conditions under which the enzyme is operating, these are considered a steady state. Kind of like Walmart, if they max out their aisles, or a grocery store, then that's going to be the steady state, right? That's going to be the rate they're going to go for the rest of that time that they have those aisles open. Okay. Under steady state conditions, your substrate's being converted to product as efficiently as possible, and so you can't actually go any faster. You're just going to continuously go until you get less substrate or enzyme. Okay. pH. Enzymes are going to be most active at their pH that is optimum. At this pH, the enzyme maintains its tertiary structure and the optimal form, and therefore its active site is going to be in the optimum orientation, optimum configuration. Changes in your pH may change the nature of the amino acid side chains and thus change that active site. And if you change that active site, you're going to either slow the enzyme down, or you may even disable the enzyme completely. Okay? And if you get these pHs far enough away, you're just going to denature it completely, like you did with a pickling something. Okay? All right. One of the reasons for this is the enzyme requires the carboxylate ion. Lowering the pH could actually change that carboxylate to carboxylic acid, no longer negative energy. It can't do salt bridges. It can't do all those things it would have done with the carboxylate ion. So again, in the body, an enzyme's pH optimum is usually based on the location of your enzyme. So for example, enzymes in the stomach tend to operate at lower pHs as their optimal environment. pHs in the as you go to, say, the small intestine, actually go up into the basic. And so, for example, trypsin here, that works on peptide bonds in the small intestine, actually its optimal pH is around 8. Okay? And here you can actually see some different pH levels. This is actually the liver. It's actually quite a basic conditions in there. And so its pH optimum is actually more like 9.7. Okay? And so it does very much depend on where in the body we're talking about. Temperature. So the temperature optimum for most human enzymes, the normal body temperature, which is 37 degrees Celsius. This is why if you go a degree or two above or below that, you start feeling miserable, right? And that's because all of your enzymes are no longer functioning optimally. They're functioning suboptimally. And if you get far enough from that, you may have almost no function at all. Okay? And for example, your brain structure will actually suffer damage if you get above, usually about 104 or so degrees Fahrenheit, right? What was the exact, does anybody know the exact number? I forget. I know you can't get up to 105. At that point, you're just gone. Okay. So again, at high temperatures, your enzyme will actually denature, but this doesn't usually start happening until you start getting into the high 40s degrees Celsius. Okay. At low temperatures, your enzyme activity is actually going to be reduced due to lack of energy. And so the reactions are going to start slowing down. The other thing that can happen is if you start freezing enzymes, you can actually start breaking their structure. Okay. As the ice crystals form, they can start shearing. So temperatures, because enzymes are major culprits in food spoilage, what do we do to food? We refrigerate it, which is reducing that temperature down into like the 15-ish, usually 10 to 15 degrees Celsius. Or we freeze it, and freezing is 0 degrees Celsius, right? And so that's how we keep things from spoiling. The colder it is, the slower all the enzymes of any living organisms are going to work. And so the freezer can keep things fresh, fresh, as long as they don't freeze or burn, right? They can keep them from spoiling longer, right? Okay. Colder you are, the longer you can halt spoilage. There are such things as negative 80 degrees Celsius freezers, by the way. Your typical freezer keeps things just a little under 0 degrees Celsius. Most of them would reach somewhere near negative 10. Okay. You can actually get colder, though. Some of the really good ones get really cold. I had a freezer that went to negative 20 once. I don't think it was meant to. Inhibitors. So enzyme inhibitors are going to prevent the active site from interacting with the substrate. Actually, there's a variety of different inhibitions, but essentially, you're either not going to form the enzyme substrate, or if you do form it, it's not going to be able to go on to the next steps. Okay? So it's going to lock it so it can never go to either the enzyme substrate or beyond the enzyme substrate. Some inhibitors are going to cause enzymes to lose catalytic activity temporarily. Others are going to cause enzymes to lose catalytic activity permanently, and you'll permanently modify the structure. This is very, very common in things like antibiotics, by the way. So in reversible inhibition, the inhibitor is going to cause the enzyme to lose catalytic activity. If the inhibitor is removed, the enzyme becomes functional again and will regain activity. In these cases, many of these cases, you can actually outcompete the inhibitor. So if you get enough substrate, you can actually end up still forcing that enzyme through. But not true for all. Reversible inhibitors, these can be competitive or non-competitive. And so this is competitive inhibition. This is the case where the substrate and the inhibitor will actually compete for that active site. Okay? These are the ones you can actually force the reaction through by adding enough substrate. It would be like you trying to compete for a desk. Anybody ever played musical chairs? So you're competing for the chair, right? So if you compete against one person, you got kind of a 50-50 shot, don't you? But if you compete against 10 people, who's going to win? The other 10 probably, right? Yeah. And so it's all about concentration with these. Competitive inhibitors are going to be concentration dependent. And again, it's just going to bind into that active site. And if it's bound into the active site, the substrate can't bind and can't go through the reaction, right? And so this actually blocks the ES complex from ever forming. Okay, this is non-competitive inhibitors. And so what they're going to do is they're going to bind to the enzyme. If they bind to the enzyme, then that active site can no longer bind the substrate. Okay? And so it doesn't matter how much substrate you put in here. These are just going to block that from ever forming. So you can't outcompete these ones. And these are called non-competitive inhibitors. All right. Now, this is an irreversible inhibition. And so what's going to happen in these is they're going to form some sort of covalent bond or structurally modify these enzymes. Okay? And they form typically covalent bonds. The most common of these are covalent bonds that are made in the active site. And so they will just permanently block the active site, and then they don't come back off because they're covalently bonded to it, right? And so the irreversible, you can't reverse the process without covalently modifying it again, okay? Your body does this trick quite a bit, actually. Yeah, your body does this all the time to enable and disable its own enzymes. So this is actually antibiotics inhibiting bacterial enzymes. Penicillin is the oldest example of this. You probably don't get to use a lot of penicillin, but a lot of these enzymes actually work very similarly. When we're talking about the antibiotics, there's a whole class of them that are penicillin-like. So what penicillin does is it binds to the active sites of an enzyme that the bacteria use in the synthesis of their cell walls. When the bacteria enzyme bonds with penicillin, the enzyme will actually lose its catalytic activity, and the growth of bacterial cell walls slows, and so your body can start fighting that slow growth of the bacteria more effectively. Okay? Basically, we're making it so the bacteria can't grow as fast and your body can catch up and kill it. Without a proper cell wall protection, the bacteria cannot survive, and the infection, in fact, stops, and then our body kills off what was there eventually. And so this is actually the penicillin right here. This R group right here doesn't actually matter that much, so we can actually change that quite a few things. And some of that times those actually increase or decrease the activity of this penicillin. This right here is your study guide for this materials. And we'll go ahead and go on to the next chapter. Anybody have any questions? Fantastic. So we have our enzymes. Okay.